Welcome to Mootechie, Structure of DNA. In this session, we will understand this topic step by step in a simple way. Now let's proceed to our first content slide. Here we have the introduction to DNA. The text says, DNA acts as the primary carrier of genetic information in biological systems. We have 3 bullet points: DNA, deoxyribonucleic acid, genetic information store, and found in living cells. Alright, so what does this mean? Think of DNA as the master blueprint, the instruction manual for building and running an organism. Every single living cell, whether in plants, animals, or bacteria, contains this molecule. It's not floating around loosely, it's securely housed within the nucleus of our cells. This is the fundamental molecule that makes you, you. It carries all the hereditary information passed down from your parents. Having understood its fundamental role, let's delve a little deeper. The heading is DNA is a genetic material. The text shows a flow. DNA stores instructions, the cell reads this information, and then functions execute tasks. The key words are genetic material, hereditary info, and cell function. So, this slide beautifully summarizes the central dogma in a very simple manner. DNA is the storage device, like the hard drive of a computer. The biological instructions it stores are essential for all life processes, from how your heart beats to the color of your eyes. The cell acts like the computer's processor. It accesses or reads this stored genetic information. Finally, based on these instructions, various functions are executed, tasks like producing proteins, which are the workhorses that carry out almost everything in our body. Now, if DNA is the instruction manual, what is it made of? Let's find out. The title is DNA is made of units. It says, just like a wall is made of bricks, DNA is built from repeating chemical units. The bullet points are repeating units, called nucleotides, and building blocks. There's a diagram labeled nucleotide structure, showing a single nucleotide with parts labeled phosphate group, deoxyribose sugar, and nitrogenous base, example adenine. Excellent analogy here. Just as you build a long, tall wall by stacking many bricks together, the enormous DNA molecule is constructed by linking together many smaller, identical type units. These fundamental building blocks are called nucleotides. Each single nucleotide, as you can see in the diagram, is itself a composite structure. It has 3 components: a phosphate group, a sugar molecule, specifically deoxyribose sugar, and 1 nitrogenous base, of which adenine is one example. We'll look at these parts in detail now. This slide is titled Parts of a Nucleotide. It states each nucleotide is a chemical structure composed of 3 distinct parts joined together. The bullet points list them: sugar molecule, phosphate group, and a nitrogenous base. The diagram again shows the nucleotide structure with labels phosphate group, deoxyribose sugar, and nitrogenous base adenine. So let's break down these 3 parts. First, the sugar molecule. In DNA, this is always deoxyribose, a 5-carbon sugar. It forms the central core of the nucleotide. Attached to one side of this sugar is the phosphate group. This is a phosphorus atom surrounded by oxygen atoms. It carries a negative charge. On the other side of the sugar is attached the nitrogenous base. This is the most important part, because it is the sequence of these bases that actually codes for genetic information. There are 4 different types of these bases in DNA. Together, the sugar and phosphate form the backbone, while the base sticks out like a protruding letter. Now that we know the parts of a single nucleotide, let's see how they connect to form a chain. The heading is Sugar-Phosphate Backbone. It says the backbone acts as the structural framework holding the DNA molecule together. Under backbone structure, it lists alternating sugar-phosphate and provides strength. The diagram shows a short sequence labeled sugar-phosphate backbone, with bases adenine, thymine, guanine, and cytosine attached, and base pairs AT and GC indicated. Okay, focus on the sides of this ladder-like structure in the diagram. This is the backbone. How is it formed? The phosphate group of one nucleotide bonds strongly with the deoxyribose sugar of the next nucleotide. This creates a long, continuous chain of alternating sugar and phosphate units. Think of it like the side rails of a ladder, strong, sturdy, and repetitive. This backbone provides structural integrity and strength to the entire DNA molecule. The nitrogenous bases, which we see labeled as A, T, G, and C, are attached to each sugar and project inward from this backbone. They're like the steps of the ladder. Speaking of the steps, what exactly are these nitrogenous bases? Let's look at the next slide. The title is Nitrogenous Bases in DNA. It says there are 4 types of nitrogenous bases found in DNA. The slide displays them: A for adenine, T for thymine, G for guanine, and C for cytosine. Very important. DNA uses a 4-letter alphabet: A, T, G, and C. These are not random. They are categorized into 2 families based on their chemical ring structure. Adenine and guanine are double ring structures called purines. They are larger molecules. Thymine and cytosine are single ring structures called pyrimidines. They are smaller. Remember these categories, purines and pyrimidines, as they are crucial for understanding how DNA pairs up. The specific order or sequence of these 4 bases along the backbone is what writes the genetic code. AGCT is different from TCGA, just like changing letters changes a word. But these bases don't just float around, they have specific partners. This brings us to a fundamental rule. This slide explains the base pairing rule. It says specific nitrogenous bases always pair together due to their chemical structure. It shows 2 pairings: adenine A pairs with thymine T via 2 hydrogen bonds, and guanine G pairs with cytosine C via 3 hydrogen bonds. This is one of the most critical concepts in molecular biology, Chargaff's rule, or the complementary base pairing rule. It is very specific and consistent. A purine always pairs with a pyrimidine. Look at the sizes. A large purine, either A or G, always pairs with a small pyrimidine, T or C. This maintains a uniform width of the DNA molecule. Chemically, adenine forms 2 hydrogen bonds specifically with thymine. Guanine forms 3 stronger hydrogen bonds with cytosine. This specificity is key. It means if you know the sequence of one strand, say ATGC, you automatically know the sequence of the opposite strand, TACG. This is absolutely vital for DNA replication and for the molecule's stability. With this rule in mind, let's visualize the complete ladder. The title is DNA Ladder Structure. It states, before we twist it, imagine DNA as a simple straight ladder. It identifies the sides as the sugar-phosphate backbone and the rungs as the nitrogenous base pairs. The diagram labels the bases A, T, G, and C, the base pairs A, T, and GC, and the backbone. So let's put it all together. Imagine a straight, untwisted ladder. The 2 upright side rails are the sugar-phosphate backbones we discussed. They run in opposite directions, which we call anti-parallel. Now, connecting these 2 sides are the rungs. Each rung is made of a pair of nitrogenous bases held together by those hydrogen bonds according to the base pairing rule. One base from one backbone and its complementary partner from the other backbone form one rung. Therefore, the rungs are the AT and GC base pairs. This ladder structure gives us a clear 2-dimensional view of how the components are organized. Now, in nature, DNA isn't a straight ladder. Why? Let's see the next concept. This slide introduces the double helix structure. It says, to save space and protect the genetic code, DNA ladder twists into a spiral shape. It has points double helix, twisted form, and maximum stability. There's a note saying, nature's perfect design. Exactly. In a cell's nucleus, there's an enormous amount of DNA. If it were a straight ladder, it would be meters long and impossible to pack. So, to achieve efficient storage, the entire ladder twists around its central axis, like a spiral staircase or a twisted rope ladder. This coiled structure is called the double helix, double for the 2 strands and helix for the spiral. This twisting isn't just for packing, it also provides tremendous chemical stability and protects the precious genetic code, the sequence of bases on the inside, from damage. It truly is an elegant and efficient design. This famous model was proposed by two scientists. Let's acknowledge their work. The slide is titled Watson and Crick Model with the year 1953. It says, the structure of DNA was famously described as a double helix by two scientists, marking a revolutionary moment in biology. It names James Watson, an American biologist, and Francis Crick, a British physicist. There's a quote, "We have discovered the secret of life," attributed to Francis Crick. This was a landmark here. James Watson and Francis Crick, using critical X-ray diffraction data from Rosalind Franklin and others, proposed this three-dimensional double helical model for DNA. This wasn't just describing a shape, it immediately suggested how DNA could replicate itself and pass on information. The quote, whether exactly said or not, captures the monumental importance of this discovery. It laid the foundation for all of modern molecular biology and genetics. Understanding the structure helps us appreciate its function. Let's see why this shape is so important. The slide connects form and function. The specific double helical structure directly enables its key functions. First, information storage. The sequence of bases hidden inside the helix is protected. Second, replication. Because the strands are complementary, they can separate and each can serve as a template to build a new identical partner strand. This ensures accurate copying of genetic information when a cell divides. Third, stability. The strong sugar-phosphate backbone and the hydrogen bonds between bases, along with the twisting, make the molecule chemically stable for a lifetime. This stability and replicability are essential for all life processes. To wrap up our understanding, let's look at a visual summary. This is a structure of DNA visual summary. It shows three stages. Stage 1, single nucleotide unit with parts phosphate group, deoxyribose sugar, nitrogenous base. Stage 2, DNA ladder structure showing base pairs and backbone. Stage 3, twisted double helix. Perfect. This brings our entire lecture full circle. We started with the smallest unit, the nucleotide, the brick. We then saw how many nucleotides link together via their sugar and phosphate to form the backbone, with bases pairing up to create the straight ladder structure. Finally, we saw how this ladder twists upon itself to form the compact, stable, and functional double helix. From a single chemical unit to the magnificent molecule of heredity, that is the structure of DNA. I hope this step-by-step explanation has made the concept clear. Remember, this is the foundational language of life itself. To explore more such fascinating topics explained in a simple, engaging manner, do check out other courses and materials on the Mootechie platform. Keep learning and keep questioning.